You're listening to the FaceTime with Scientists podcast, where we go into the lab or into the field to learn from scientists about their lives, their work, their struggles, and their joy. Let's go. Awesome. Well, today, everyone, I'm here with Taylor Hailstock. You are a PhD student, or actually, you recently defended your PhD. So, congratulations at Emory University. And I'm super excited to talk to you. I think I've seen on your Instagram, I was particularly interested when you talk about dissecting brains and looking at, yeah, how brains work. And we'll get into all that. But can you tell us a little bit about where you are today and what you're doing?
Yeah, so I'm in my lab right now. And like you said, I did recently defend, but I'm in this like weird transition phase where I really do want to finish the projects that I was working on and send them out for publication before I move on to my postdoc. So I'm still in
Gotcha.
a lab in the Department of Cell Bio at Emory.
Nice. Yeah, I was a little when I saw you defended your PhD. Some people are like, "Okay, done with the lab work," but that's cool that you're getting to finish out the project.
Yeah.
Very cool. Well, what do you have you're at the scope, like, what do you have what are you looking at today?
Yes. So right now I'm dissecting larval brains. So I have my flies here. I picked the ones that are like crawling stage and literally use like a pointy tool and just go behind them and pick them up, put them on my slide. And as you can see, they're like really small. So I'm just using a regular tabletop microscope to magnify them a little bit so I can more so see what I'm trying to get out, which is the brain.
Gotcha. I mean, that must be really so you're that's when you say fly, it's like a fruit fly, right? Like
Yeah,
Drosophila. Yeah.
yes, exactly. Drosophila
Droso
melanogaster. So we don't use like any fancy types of flies, but like the basic fruit fly.
and I imagine that must be very difficult to have a tiny fly dissect just the brain. So are you when you say dissection, you're having to just get the whole brain out of the fly?
Exactly. And like since I just put that one down, I took it out. So I've gained a lot of speed by doing it for a long time, but it really is difficult because it's all transparent at this stage. Like once they get older, like you can see the abdomen, you can see the eyes, but at this stage it's kind of just like a worm and it's completely transparent. So the magnification definitely helps. And then just like with anything else, training yourself and looking at textbook examples online, what it's supposed to look at, and then figuring it out on the scope.
How long has it taken you to, like, get really quick at this? I mean, how long have you been dissecting flies?
So I started my PhD in 2021, and I'm in the biochemistry, cellular, and developmental biology program, or I was. And we do 3 rotations. This was my last rotation. And I would say from like February to May, the entire rotation, it took me that long to be comfortable with dissecting. And then now I've just become more comfortable with like getting a clean dissection every single time.
Right. Well, what does it look like? I mean, does it you can see like, relief of the brain, or is it just, like, how do you recognize the brain in there?
Let me see if I can set us up so I could show an example. So this is one larva that I just put down here. And what you see me using are just like regular tweezers, nothing special or fancy. Try to change the focus. Put it away. And so yeah, um, as you can see, it's still moving.
Right.
So I snip the back area to kind of like immobilize it. But the part that you still see moving, like right here, are like the mouth hooks. So that's the head region. And the brain should be somewhere right here. So my technique is to like snip here, kind of like this, open it up, and then hopefully it'll flow out.
Oh.
So let's see if I can actually do that. The screen that you're looking at is also like inverted from what I see.
Oh, that's okay.
So bear with me. Okay. So I'll hold it right here. Snip. And then I'm basically trying to like tease out what's right here without bringing other stuff. Hang on, so I'm gonna hold it right here for a second. The brain looks like two Mickey Mouse ears and a tail. And so in this glob, it's right here.
Wow.
So it's really hard unless you know what you're looking for.
Or
But
yeah.
we got textbooks and all of that. So I know that what I'm looking for is one lobe. The second lobe is somewhere under here. Next to this are wing discs. This might be the other lobe, and that is the ventral nerve cord. So this altogether makes up the fly's central nervous system, and that's why we use it as a model. But yeah, and then so what I just did was gross dissection. You would then go back through and like get these things off and just have a cleaner brain.
Right.
So today these are going like for say immunofluorescence experiment or maybe like sequencing. And based on what you're doing, it may or may not have to be as clean.
Right.
But yeah, I think
That's
you've probably seen on my Instagram me doing adult brains. So I'm gonna put an adult fly down here.
oh.
So you can see the size difference
Right.
like immediately. But the brain in the adult is way more complex. And then you also have, color. You can also see the segments. You have wings. But all of
Right.
this is started here. So the cool thing about fly is you can kind of look across development and it won't take that long to observe the system.
Right.
And yeah, so for adults, I would start with the head. I'll take this one away. And here, that's where things get tricky because you could tell already I kind of like dented it.
Right.
Yeah, and hopefully it doesn't float away. Okay.
Yeah, talk about fine motor control.
Yeah, and honestly, one of the top comments I get is like no gloves. Really, the flies aren't like hazardous to us. But on top of that, like you just mentioned, you need as much dexterity as you can get. And I think the gloves would take a little bit of that away from me personally.
Right.
Uh, Put it back in center. And yeah, that was really quick. I think I was just lucky just then. But as you can see with the adult brain, it's like still wrapped up in so many other things now that the fly has eyes, it has like cuticle, skin, little hairs. So it's not as straightforward to see. Let's see if I could clean this up real quick. Okay. And this is kind of what the adult brain looks like. So once it grows from like the two Mickey Mouse ears in the tail, it kind of spreads out horizontally and it's just way more complex. So that's kind of what my day to day starts with, no matter what type of experiment I'm doing, but it will all start with a dissection.
Really? How many do you usually have to dissect for like a single experiment?
Um, if I'm doing like a staining, my N is usually 10 per replicate, but then we always try to do things in triplicate, to be as rigorous as possible. And then say like for RNA immunoprecipitation, things like that, we do 50 brains per replicate. And one of our techs, I'm working with her on getting a data set for sequencing. She's doing 150 per genotype per
Wow.
replicate. So, you just have to scale up based on what type of data you want to get.
Right. That's fascinating. Well, to back up a little bit, so I mean, I have a variety of questions, but one is just like, why are you looking at fruit fly brains? Like, what are you actually studying in terms of brain and development over time in the brain?
Yeah, great question. And honestly, the most important question. I guess we should have started there. But the big picture question for me, my project, and kind of where I think a new direction of our lab is going is how does the brain know how big to grow? And so I'm studying, of course, we know the central dogma that it's DNA, RNA, protein, but I'm looking more at the post transcriptional regulation level, and I'm looking at an RNA binding protein, and more so just asking the question, say in neurodevelopmental disorders where the brain size is messed up, you have macrocephaly, which is when a human baby is born with a brain or head that's too big, and you have microcephaly when the brain and the head is too small. What's going on molecularly? Like what mechanism is messed up that causes that to happen? And so I'm using fruit flies because there are a lot of conserved mechanisms between flies and humans. I know that sounds really crazy. And I'm not saying that like a fly brain is the same as a human brain, but when I'm asking questions about neurodevelopment, it makes most sense to me to start at the neural stem cells, the cells that are driving all the other cell types that come. And when you look at a lot of the mechanisms in terms of how neural stem cells are regulated, how they divide, um, making neurons and glia, those types of cells, a lot of it is just the same in fruit flies. In terms of logistically, we know that like mice, bigger model organisms, like I used to work with pigs in the government, they have to be so highly regulated. Like you have to get approval. They tell you how many that you can have. Flies, you see that, I'm just growing them up. They also have a 7-day life cycle. And I would say the biggest reason to use them is when you're asking questions about something over neurodevelopment, you kind of want to be able to track it and see it over development. Just like I showed you the larva, I can watch, say, a fly grow up and I can look at the consequences of some things that I can easily change genetically.
Yeah, that's fascinating. Yeah, I think, there's a lot of well, there's been some, right, I guess, news about science, right, where people are like, "Oh, these scientists are studying flies, why
Yes.
would that be important, right?" Like, but then, like you point out, there's so many things that the fly I mean, ultimately, you would go on and study this maybe in other organisms, but in terms of, like, tracking like you said, they develop quickly, so you can track a whole organism and replicates, many replicates so that you can do that. I mean, that would be difficult to do with larger animals or take a lot of time. So just but do you have, like, other answers that you give people when they're like, "Why flies?"
Just that, honestly. And I would say, in terms of from a geneticist perspective, we don't necessarily have to send our sequence, get a fly injected in order to express something differently. Like the pipeline in terms of getting experiments done is way shorter. For example, the flies that I'm working with, you saw that they had red eyes. Even just introducing an eye color, like fly geneticists are so cool in that they created balancers. So I can tell whether or not my gene is expressed fully if it's only half of a dose just by things like the eye color, if the wings are curly, if they have short hairs on their torso instead of the normal length of hair. So yeah, I would say all the things that we run into when you're designing experiments, you want to know whether or not your controls work, things like that, is so much easier using flies.
Yeah. Yeah, that's super cool. Well, yeah, so I had a question about, yeah, in terms of, like, studying how the brain grows. Do you study particular diseases? Like, I know, like, what is it, Zika that we heard a lot about, um,
That's a
infectious,
question.
diseases that yeah.
When we started the project, we were just focused on brain growth in general because like you saw, you can kind of predict the size that the fly is supposed to be. And when we knocked out this gene, it's called ORB2. It's a RNA binding protein. We actually were looking at the other version of it, which is just ORB, and it's expressed like in the germline. That's what my lab focused on. Like the female germline and oocytes, the ovaries, things like that, and RNAs there. ORB2 was more highly expressed in the brain, and then literally looking under the scope when you dissect your first brain, that's a ORB2 null. So when you don't have the gene, it was dramatically smaller. And so then we started looking up, okay, what human developmental diseases or disorders, that's the same phenotype. And so mainly all we're looking at is microcephaly. But like you mentioned, Zika virus, microcephaly can be like genetic, but it also can just be seemingly randomly happening. And so we're kind of looking not at the known exposure to things that can, like Zika virus, that can cause pregnant mothers to expose themselves and then therefore their babies to the potential of microcephaly, but more so looking for the genetic answer. Um, and then also our protein ORB2, it is orthologous to a human protein that has been clinically noted to be like deleted, mutated in human microcephaly patients. So that's kind of our clinical relevance, our link right now. And so, what I think would be a cool direction from what I found is to insert the human version of the same gene into the fly and see do the same things happen. And then that would let us know, okay,
Mm,
maybe that could be something we add to like the prenatal screening. Maybe that's something to look at once we do see any like inclination that a baby might have microcephaly while it's still in Europe.
that's really interesting. In terms of the connection there with the virus, does the virus, Zika virus, for example, like things that cause environmental causes of microcephaly, would it change genetics or maybe that would be a different mechanism? Or can you still learn about those by studying like these genetic causes? And then would that just be like a mutation in the mom or a mutation that just like happened to be in the egg that would like cause this, the orb to, you said, to like change and cause this?
I really don't know. I think it could be everything. I think as I've gotten more experience, I've also just realized that the possibilities are endless and none of the things are like mutually exclusive. All of the things could be happening
Right.
and causing things at the same time, which is honestly what keeps me interested in science. I think if things were just black and white, it would be less people with breakthrough discoveries and less jobs available, less projects for people to work on. But I really don't know. I do think and believe that, like genetics and environment play a role, but I don't think that they're equal. I don't know how the scales tip though, realistically.
Right.
You know,
Right. That's super cool. How did you get interested in this? I mean, have you always been interested in the brain and then this idea of like how the brain knows how big to grow?
um, I would say I wasn't at all interested, like say specifically in the brain. My kind of through line of interest has been in neural stem cells. Just because I thought it was so
Mm,
cool that something that is a cell itself has a role to control other cells, you know? And I think looking at the
right.
brain from a molecular standpoint, it's more so about transcription factors and how, like, say, we could use the analogy of DNA itself being a cookbook. RNA, you could have like all these different recipes, but there's still some type of checkpoint. There's still some type of master regulator that decides, just like we decide, okay, which recipe am I going to use today? And then, like, whether or not the protein is expressed or not, or even just how it's expressed, even can be changed, like different isoforms, stuff like that. The protein would be the meal that we actually make. I think looking at it from the question of how are these things playing together, what controls what, has made it most interesting. But I wasn't at all always interested in the brain. mainly just interested in neural stem cells and regeneration. Like, what can they do, and how can we as scientists like take advantage of them, exploit them to help heal things, to help repair, and to like answer questions like this.
Yeah. Oh, that's so fascinating. Yeah. And can you explain a little bit of, like, what you explained a little bit, but, like, in terms of transcription factors, like, what are those? And I know you said you like orb-2 being an RNA binding protein, so I know that some of those, like, will bind the RNA to, like, take them somewhere that they need to be or bind it to in order to like, what is it yeah, what is it doing to the RNA then, I guess?
That's a good question. So for me, I'm mostly interested in transcription factors that are like mRNAs. And in the fly neural stem cells, sorry, I have one larva and it's just moving around. I forgot that it was there. Um, yeah. So mainly the ones that we're interested in are expressed in stem cells, and the fly neural stem cells they divide asymmetrically, which is in design to make two different fates. So the neural stem cell can self-renew, which is one of the classical like definitions of a neural stem cell, but it can also make a daughter or start to make its progeny, which are usually glia, neurons, all the other complex cell types. And so a transcription factor, for example, would be something like in flies Miranda. It's expressed at a certain pole, so the apical pole, not the basal one. And that asymmetric division will like segregate it from being near the cells that are going to differentiate and make sure that it's only at the top where the neural stem cell will self renew. And so ORB2 or other like RNA binding proteins, there are a lot that are like implicated in FMRP, Fragile X syndrome, other neurodevelopmental disorders. And they do exactly like what you said. They bind the mRNAs. Ours specifically bind at a certain, like a canonical, a conserved site, which is how I know that the human ortholog does a certain thing because they all share a certain
Mm,
sequence at the beginning. So they'll find the mRNA. Like you said, the RNA, they're kind of just everywhere. And especially during development, it's a highly regulated process, or it should be, so things don't go wrong. The RNA binding protein will recognize that site, it'll bind to it, and it can either cause its immediate, like local translation. So, hey, this fly is 4 days old. It's about time to start making wings. I'm going to allow the wing transcription factor or gene to now be expressed. But now we're getting to day 6 and we need to focus on, um, say, the eyes coming in. The RNA binding protein might bind it and stop the translation. It also can transport the mRNAs. Some mRNAs should not be expressed in a certain area that will like become a certain organ. So what I'm looking at are genes that should be expressed in the brain. And for example, like in our genetics classes here at Emory, we show this example that if you overexpress or like artificially, express a gene, say in the thorax, you could make eyes come in the thorax. And that just speaks to how tightly regulated this has to be. It's spatial, it's temporal, it's really complicated.
right. Yeah. It's I mean, I've always been fascinated in, like, yeah, I guess neurodevelopment, that process. I don't know a whole lot about it in terms of, like, I haven't really studied it. I didn't study it in school, but just how complicated that is and, like you said, all the gene expression, like, changes from, like, moment to moment or from spatially, like, it shouldn't it should be translated here or not here, and how much that's important to, like, forming yeah, to, like, forming the embryo and making sure things are where they're supposed to be, I guess. It's
And the fact
crazy.
that even when things go wrong, the organism is still alive. Like for me, that was one of the things I was
Right.
surprised at. Like I see the brain is smaller. Even think about humans. A lot of babies who are born with microcephaly. Someone visited our lab recently and he has a sister who's 4 years old and she has microcephaly. Things that you would think conceptually would be fatal. The body is so, yes, it's so like interwebbed in terms of the processes, but some things must be individual that you overcome and that you're able to still have a life, like say whether it's an organism like this or as big of an organism as us. So I was really captivated by that, and I think that further motivated me to want to know what exactly is happening.
Yeah, that's fascinating. Well, in terms of, like, trying to what you study, is it kind of just trying to understand the process of this? Like, are there any downstream applications that you're working on, or is it just, like, you're trying to understand this so that others could maybe know what to target or that kind of thing?
I think we're really just at the beginning. Like, this is the first project that studies ORB2 in the brain. Other people have looked at it because it's important, like for transcription, development. And that's the cool thing about using flies too. Almost everything people already studied, like in the 80s, early 90s. But I think, yeah, this is more so the first breakthrough in terms of relating ORB2 to the phenotype of brain size. And I think that speaks to something like our workflow as scientists. We're starting at the phenotype. Everybody doesn't necessarily do, I guess, top down approach. We could have started like at the molecule and looked at, okay, where does the molecule go? What does it do molecularly? And so, yeah, I think we're at the point where we just have a phenotype and not necessarily an explanation. So I wanted to share like my thought process. When I first started, remember I told you all I saw was a smaller brain. I could think of like three
Right.
scenarios that were happening. One is everything made correctly and everything's just smaller. Like that could be possible. Or is something not being made? Like is something missing from the beginning? Or are things being made correctly and then like somewhere during them being made, something happens? Like, are the cells dying? Are the cells over dividing? And then like you're doing too much and so each one is less perfect. They're smaller, they're more
Mm.
prone to mutations. I think we're just still at that point of figuring out what's going on. But even where we are, at least we've identified a molecule that we could start to apply downstream as long as we link it back to humans. So I think doing the experiment of putting in the human version, seeing do we get the same effects, will kind of give the rationale to like go forward with the downstream.
Right. Yeah, that's so interesting. I mean, just in saying microcephaly, right? I mean, which just means small brain, right? That like even if you diagnose someone with that, like, all that means is it's just that we see the phenotype of a small brain, but you don't necessarily know. I mean, is it true that in most cases, I guess, of microcephaly currently that happen in the US, like there's not necessarily, we don't necessarily know why it happened?
Yeah, I've never been in a hospital room when it's diagnosed, but one thing I love about now sharing my science on social media, a lot of people who are microcephaly moms or know someone who have microcephaly or have microcephaly themselves, that's what they let me know that like all they say is exactly what you said. Hey, you have a smaller brain. And it's like, yeah, I can see that. Yes, I know that something is going on, but no, I don't think we have a molecular explanation for it outside of those environmental exposure cases. The link with microcephaly and Zika virus has
Right.
been highly studied. But, I just feel like I know I would want more of an answer, whether it's how it happened, what can I do, or what can I tell other people to look for, like say in some of these screenings, to just be more informed.
Right. Do you really enjoy do you enjoy the type of research you do in the lab now, and are you hoping to continue that? Like, what are your plans? I guess,
Yeah,
yeah,
good question.
going
I've
forward.
actually a lot of different things so far, and this is by far my favorite. I started out doing pig research, which just wasn't what I wanted to do. I was separating pig blood and like using flow cytometry to look at different cytokines. It was more like an immunology project. Then,
Uh,
I mentioned doing those rotations. I rotated and I did a project using mice and looking at stem cells, which was closer to what I wanted, stem cell stuff. But I think for me, flies are the optimal model organism. I definitely wanted to continue either using cells or like an organism where I can isolate cells. But I think what was most interesting during my time in my PhD was using organoids. You can do fly organoids,
yeah.
and so Yeah, I'm interested. I'm going into a postdoc, and I will want to continue studying like molecular mechanisms and connecting them to development, to diseases. But I'm looking forward to like new questions, new systems, new approaches.
That's cool. I didn't realize, I guess I should have realized it, that you can make, like, fly organoids. What would
Yes.
be so I know, like, in general, the idea of, like, making organoids, right, like, I thought of, like, okay, well, we can't study the whole human brain, but we're, like, isolating cells and growing, like, little mini models of the human brain in a dish. If you were taking, like, flies and creating a fly organoid, like, what would be the purpose of that when you can study the whole brain of the fly?
I think one way I think about it is once you like take the brain out, so like the brains that I have sitting here on this slide, they start to decay immediately. And you
Oh,
could
right.
study the brain, but like you can study it while it's still on the body, you know what I mean? And once we take them out, we have to either immediately fix and then look at the fixed tissue. You could also live image, but like as the time goes, you're losing the quality. So the way I think about it,
Right.
we'll kind of be able to like suspend the specific organ that we want. There's also, I think it's called expansion microscopy. You can also like magnify what you're looking at. And I think that could be really cool to not have to always look under the scope because you know the lights, it gets hard on your eyes and stuff. But yeah, I think maybe mostly for isolating what you want and being able to observe it longer and then also being able to manipulate it easier. You know, like I've explanted the brain, but like I still have to take them, put them into a tube. I still have to be careful, like taking the liquid in and out the tube. And I really do lose a lot of brains. That's why I usually over dissect. So I think having them kind of like in a controlled matrix would extend the amount of time that we're able to look at them.
Right, that makes sense, yeah, I didn't think about that, that, like, yeah, once you take out the brain, it's no longer living, but you can make an organoid from the beginning and then change exactly when you want cells to have a mutation or that kinda thing, and then
Exactly.
I mean, I interviewed Nuri, and she has, like, organoids that literally, like, brain cells are, like, sending signals to muscle cells, and she can study, like, the signals and where, like, motor neurons are connecting to muscle tissue, and you wouldn't be able to do that, like, I mean, in a brain that you've dissected because it's no longer, like,
Add
active like that.
to it. Exactly. But I haven't done it yet. So I hope that it will work, but that's something I would definitely be trying.
Right. That's super cool. What about, like, challenges? Do you have, like, things that I don't know. Well, I mean, you just defended your PhD, so, like, that's right, like, amazing. Like, you did it, you're done with that project, but were there any was there anything really difficult that you had to, like, troubleshoot along the way? Or things that are facing you that are difficult now that you wanted to
Yeah.
talk about?
I don't know. I don't know which thing to choose. I would say something that I think I've worked through so far or I'm still overcoming is one, just not the imposter syndrome in the sense of like, do I belong here? Am I capable type? But when it comes to like actually designing experiments, so like doing genetic crosses and stuff, you really have to trust that you do know what you're doing. And I think for a long time, I was second guessing. I would, you know, like say you're doing an experiment, you're following a protocol, but I would think twice about stuff like, but do I really need to add this? I think becoming just more on autopilot, it takes out some of the bias that we might introduce by just being unsure. I think that's something that we don't talk about. Like we talk about bias because you don't like blind your images and you might already know what you're looking at, stuff like that. But I think the way that we're personally feeling and like our attitude while doing the research could also cause some bias in the sense that we double guess ourselves or like we backtrack when actually it's okay to just follow what the protocol says. And if it doesn't work, all you could do is throw it away and start again. So
Yeah,
that, and then I think just working with model organisms is kind of difficult because you don't know until you compare the actual controls like what I was saying, what's a phenotype and what could just be because, oh, is it that the flies are in a vial and they're just not happy? Like there's just so
Mm,
many different factors. And I think as a scientist, yes, we're the authority, in the equation, but we also have to be open to seeing things for what they are, which is at the end of the day, still like an organism, just like us. Yes, flies do get overcrowded, and that is a factor that we have to consider in terms of whether they're growing or not. They do compete for food, resources, just like us. I think we have to be quite empathetic, but then also get the job done in terms of dissect them at the end of the day.
right. No, that's so fascinating. I mean, do you find that yeah, like, how did you kinda overcome that, like, uncertainty? Was it just, like, more practice and more doing it, or do you have, like, tips of how to, I don't know, move
Really,
beyond that?
I think more doing it and realizing no matter what I do, if it was going to fail, it was just going to fail. And then I think over time of feeling like you're trying your hardest, you're staying late, you're not listening to music because you're trying to focus, it doesn't change the outcome. And so you might as well be comfortable. You might as well say wear a pink lab coat if you feel like it, create like more of a space where you feel comfortable. And then you'll be more confident. Okay, nothing that I was feeling contributed to how this experiment went. Like, this is not at all personal. These are just the data, and we can move on from it. So I think honestly, the only way to overcome is to keep working through it. And then you, as a person, honestly, will get tired of feeling like it's your fault. And then you'll consider,
Right,
okay, maybe it was just the experiment. Like a
right,
negative result isn't always because you did something wrong. At some point, we have to get enough to even like say the example I gave. I had to test, are the cells bigger or smaller? Are the cells dying? Are the cells dividing when they shouldn't be? And even though all of those things I got negative results, it led me to find the actual answer. But it wasn't a result of me not knowing what I'm doing, not doing it well. It was just the data.
right. No, I think that's so key because, yeah, I mean, those of us I'm the type of person who would take things very personal, right? Like, if it's not done if it didn't work, I'd be like, "Well, what did I do wrong?" Right? Where it's like, yeah, you kinda have to trust, like I mean, no results are just as important as something actually happening in terms of, like, leading you to the answer and just, yeah, trusting that you observe and what comes back is not a reflection of how well you did at it, but it's just, like, yeah, what it's telling you. Well, thanks so much for telling me about your research. Do you have anything else that you kinda wanted to show or to kind of explain about your day-to-day process?
Mm, not really. Just that, day to day, we're definitely in here maintaining the flies. We have to feed them, keep them happy, change them around. But I think something I'm seeing reoccurring, like say in my comments, is just the big question of, but why though? And like
Right,
questioning the ethics behind using an animal model. But like what we just talked about, there is no possible way for me to take a human baby and test out why they have microcephaly. And I
right,
think I said in one of my latest videos, I just really do think as a young scientist, my philosophy has started to be shaped by the rate of science is really going to depend on how creative we're willing to be. And I'm a person who's willing, yes, I'm willing to like touch flies and work with them, but like all the new technology, say like organoids, things like that, there really are some creative ways to go about science instead of waiting another 20 years, like say for this generation of people who may or may not want to donate their bodies to science. And with everything going on, there's a lot of misinformation. I think, especially with that going on, we might not have as many samples to even push, the rate of research. So just want to highlight that model organisms are very much necessary, and because things are conserved, I think that's the cool thing about, development, evolution, all the things that we learn about in school. We can move backwards, like backtrack from human and like, okay, if this was still here in a fly, maybe let's use this organism to try to get some more info. It doesn't have to halt the progress of discovery.
right. Yeah, it's so true. I mean, I think we're on the cusp, right? Like with organoids and different things of just, like you said, being able to do a lot, potentially a lot more science with a lot less. Um, and being able to answer some of those early questions, like you said, you can't manipulate, well, sometimes I think, let me just imagine larger animals, much less humans, like, you can't manipulate them to make small brain so how do you tell what's happening unless you use model organisms or organoids, but, like, different types of these things answer different questions, which I think maybe people don't maybe think about as, like, we could answer something in an organoid, but if we don't need the whole organism, maybe a fly to answer something else, and then we can combine all that knowledge to say we're confident in this happening, higher up the chain or, like, the more organism you get, so
Yeah, and science is always evolving. Like just because this is what I found out right now doesn't mean that that is the answer. It doesn't mean that there's not more to the answer, you know? So just keeping an open mind. That's why I love what you're doing with this podcast because being able to, I think really just putting a face behind the work, it just makes it more relatable and you realize, okay, wait, this is just a person who cares about the outcome and like learning more about the development, the disease itself. And that's why they're doing all of these little things to try to get at it. Like
yeah.
humanizing science.
Yeah. Well, speaking of that, so, I mean, you maintain your own, social media account where, I think I lately on the podcast, I do have a lot of people who, reaching out to people who do social media and talk about their science, on social, and I just think that's become such an amazing way for scientists individually to help humanize science. How has that process been? Like, have you done it throughout your whole PhD? Like, why did you start it? Has it been something that's encouraged by your advisors?
Really encouraged by my friends and family. That dissecting video that went viral was my first video. I literally never showed anything, honestly, because I'm the type of person, I was just 100% locked in, to be honest. Like, I moved away from home to come down here and get my PhD. And, sometimes it feels like life stops when you're away from your family, away from your friends and stuff. And so I think for the majority of my PhD, I was more so like, anytime I'm awake, I'm working. I don't care about nothing else. I just want to make it back home and get my degree. But like now that I had the time and the wiggle room to kind of breathe, I was just looking at my images and I was like, wait, actually this is really cool. And I think there are other people who would think this is cool. My peers too, I started to notice, okay, I dress differently than other people. I present myself a little differently than other people. And I wanted to show people, and that's why I haven't made like a science page. It's just my regular Instagram. I want to show people that you can do it all, especially young
Mm,
women. You don't have to compartmentalize. Like a lot of people ask, oh, you do this with nails? Yes, because I use my fingers to hold the stuff. You know what I mean?
yeah.
I just was randomly inspired and decided to accept, okay, maybe my peers and my family do have my best interests and they're not telling me that this is cool just because they love me and support me. Maybe this is actually
Yeah.
cool. So no, that literally was my first video. I just made a YouTube because I realized a lot of people comment that they would love to hear me talk about it. So I'm trying to get more into that. I just got my microphone from my friend. And so yeah, no, I literally just started. And for me, we also just got a teaching scope. Like that's how I showed you what I'm doing. I just didn't know how to record it. I see that some people they have like mounts that you could put on a microscope. That would be
Right.
cool. But yeah, no. I'm just starting and I think I'm finding it really good because I think no matter what how people take it, it's exposure. Like I think there are probably people who never knew that people use flies for anything. Like why would you
Right.
use them for anything? I think there's probably people who didn't know that lab coats come in different colors. Whether
Yeah.
it's regular exposure to something. And then I feel like right now it feels like a double-edged sword just because the influx of comments that don't necessarily apply to what I'm doing. Like a lot of people comment, wow, Bill Gates is doing this with mosquitoes. And I'm just like, oh, I didn't know that. And I have nothing to say about that. I was just showing my thing. But I think it at least gives me an opportunity to exercise what I've learned about science communication. And the biggest gift so far has been hearing from either people who know someone with microcephaly and who feel that me sitting here for hours dissecting brains really matters and it means something to them. And then the generations behind us who are like, wow, I want to be just like you. And I'm like, you're going to be better than me, but thank you. And I'm glad that you could see someone that you would want to aspire to be like doing something so serious as this. You know, like I feel like scientists, even like say in the new Spider-Man movie, the last
Yeah.
Jurassic movie, I feel like they're always portrayed as a certain person. But when I look
Oh,
around my
yeah.
lab, that's not what I see. And I just want to show people that.
Oh, yeah, absolutely. It's so important, and I love so much what you said. I think that, like, yeah, just showing people what science looks like, what it can look like to be a scientist, and breaking some of those stereotypes too is so important, and I think you're doing the the good work. Do you get I mean, I'm sure you get there's a lot of positive, but is there any, like, negative in that you have to, like, struggle through in terms of, like, sharing your science so much on especially on social?
I would say one thing I'm learning, or, it really affected me being in this environment. I think in the scientific environment, a lot of people use their knowledge like as a weapon to make other people feel dumb, to make other people feel like they don't know what they're talking about, or you should know this, don't you know that? And that's something that I feel, say, a lot of the professors when I was still taking classes, I felt that even making it to this level. I got a master's, I did my bachelor's, and I was like, wow, I didn't feel dumb until you said that. So I think now that I have this wealth of knowledge, we as scientists, it's like a balance. We have to learn when to respond at the high level, when to not dumb things down, but I think it's reimagine how we explain it. And honestly, that's the test of whether you know what you're talking about or not. If you can explain it in different ways, like using analogies, just being a regular person about it. Um, so in terms of the negative comments, I try to use them as like an exercise for myself. Like, okay, how can you respond to this in a way where you're ignoring the negative aspect of it? Like, say if someone is saying something about me, something negative in whatever instance, or a lot of people question like the PPE that we're required to wear working with flies. How can I turn this
Right.
into a teaching moment? And me as personally, that's the type of person I want to be. I'm a person who's very open and willing to engage in confrontation, because I think a lot of things you do need to say the quiet things, the unspoken things out loud, and bring the room to a balance, call out the elephant in the room. But I don't want to be malicious with it. So nothing that's too negative that I haven't been able to try to rework and work through. And really it's just, I think it's the lack of exposure. We don't show scientists doing anything other than wearing full PPE. And that's our fault.
Right. Yeah, I think that just, like, that transparency is so important in science, right? And then explaining the context for these things. I think that speaking of everything's going that's going on right now with COVID and
Yes.
the, like, looking back at what happened and how it was dealt with in terms of recommendations, to me, I just think that there's like, I know that scientists, by and large, right, as a group, I mean, like, we're humans usually with a mission that we care and, like, we're trying to make the best possible, say what we know, say what we don't know. But I think when it comes to that public facing, like you said, there could be people that come back and have questions, and you're like, well, how do I handle this? And I think it can, I mean, you're human. It can get like to where you're kind of defensive or you're like,
Exactly.
you're wanting people to follow maybe a specific recommendation, but just being really transparent and explaining the context for like in, PPE that you would need it in this context in particular, but in this one, like it's okay. Like, and just like trying to explain that complexities of that, and humans were not great at nuance in general, but right? But I think it's really important to kind of explain those things. So that's really
Yeah.
cool.
Yeah, I think we could do a much, much better job. I actually went to school, to grad school initially because I wanted to go into science policy. I'm from Maryland. Both of my
Oh,
parents just retired from their whole life in the federal government. So for that to actually be my original goal, and then everything that transpired while I was getting my PhD, for me, it really was an eye opener. And I definitely want to return to it. I just don't think now is the best time for me. Maybe that's like a settle down job for me, like have a desk, not be at the bench, that type of stuff. But
right.
really do think the issue is communication. I think
Yeah,
a lot of like those buzzwords and stuff, like gain of function. Why is that a buzzword when actually that's what we do and study? I think we just could have gone about things or put more effort into understanding where the public was coming from and how they
right,
understood things originally.
right. Well, thank you so much, Taylor. It's been a joy chatting with you, and seeing your expert skills at dissection is, yeah, fascinating.
Thank you. I was so happy to speak to you too. And again, I really love what you're doing. I think this really does feel like a FaceTime with a scientist.
Awesome. Yeah, thank you so much.